Good afternoon and welcome to the USDA Human Nutrition Research Center on Aging's Monday seminar series. Before introducing today's speaker, let me remind you to ask your questions for our speaker using the Q&A icon located on the webinar toolbar. I encourage you to ask your questions as they arise during the presentation. Let me now welcome today's speaker, Dr. Stacy Andersen. Dr. Andersen is an assistant professor of medicine at the Boston University School of Medicine. Dr. Andersen has been investigating cognitive function in long-lived individuals and their family members in the New England Centenarian Study and the Long Life Family Study for the past 20 years. She's a behavioral neuroscientist, and her research focuses on identifying factors associated with the resilience to Alzheimer's disease among centenarians and their family members. Her research also involves the development of novel methods of detecting cognitive change using digital technologies and behavioral features of cognitive test performance. And today, Dr. Andersen will present her research in a presentation titled "Leveraging Extremes to Learn About Aging and Brain Health." And with that, let me turn the microphone over to Dr. Andersen.
Great. Thank you so much for that great introduction and for inviting me to talk here today about what we've been learning, leveraging the extreme cohorts that we study, and even some extreme measures that we use to detect brain health. So I've been studying exceptional longevity for the past 20 years with Dr. Thomas Perls. And he developed a strong interest in this field back when he was a geriatrics fellow. He had been assigned patients at this older adult living community, and he had always been taught in his medical training, the older you get, the sicker you get. So when he was assigned 2 patients over the age of 100, he, not surprisingly, imagined that they would have a lot of diseases and would be confined to their rooms. But that really wasn't the case. They actually were 2 of his healthiest patients and hardest to track down because they were out in the community doing lots of activities, very engaged. And so he wondered whether these were 2 just exceptional outliers or if there's something different about these people who live to extreme old ages. And so over the past 30 years, so he started the New England Centenarian Study, and over the past almost 30 years, we've enrolled about 3,000 90-year-olds and centenarians, ranging in age from 90 to 119 years of age. And we also follow their offspring. So we have over 600 offspring ranging in age from their 50s, and even some of them have become centenarians. And this picture that you see here is from Time magazine, and it shows the oldest participant in our study, the woman in the blue dress, who died at 119. And in this photo, the woman in the red dress is her daughter, who actually became a centenarian herself and died at age 101. And there are actually 6 generations of her family in this wonderful photo. And then in addition to the centenarians and offspring that we enroll, we also enroll controls that we use to compare to the centenarian offspring. And so what do I mean when I say that we study exceptional longevity? Well, we're really trying to look at the underlying determinants of living well beyond what's average. So in our studies, we're talking about living 30 to 40 years past the median survival of the 1900 birth year cohort. And when the study started, we were initially studying people from that 1900 birth cohort. And you can see in this timeline here that for that birth cohort, the median survival was 67 years. Only 5% of the people born in 1900 made it to age 92. And only 0.2% of the people born in 1900 made it to age 100. So there's a huge drop-off in survival, and therefore a great deal of selection that happens in those nonagenarian years. And so we believe there's a much different underlying contributors to reaching these very extreme ages. And although centenarians have been increasing in number and proportion over the years, they are still quite rare. So Only 1 out of 5,000 in the US population is age 100 or over. Only 1 out of 200,000 is age 105 or over, what we call semi-supercentenarians. And then 1 out of 5 million is age 110 and over, what we call supercentenarians. But what is the most extreme case of human aging? Well, that was Madame Calment, who died in 1997 at the age of 122. So she defines what we know as human lifespan, the longest recorded lifespan. And she's really the only one on record to have broken through or surpassed that 120-year mark. And here she is at age 116. She was sitting bolt upright and engaged in the conversation that was going on with the photographer. And she has a very interesting history. She took up fencing at age 85. She rode her bike until age 100. She lived alone until age 110. And she really only moved into a nursing home beyond that age due to poor vision. And she actually had no illnesses until the age of 114 when she had a hip fracture. And here's another super exceptional case. This is our oldest participant that I mentioned earlier, Sarah Knaus. She lived to 119, the third oldest ever. But in this picture, she is age 118. And here she is at age 99. So even just looking at these pictures, it seems like these people with exceptional longevity have a different aging trajectory. They even appear to be aging more slowly. And so over the years, we wanted to look deeper into that. And so we looked at the onset of their first age-associated illness, the ones that you see listed here. And we noticed that centenarians didn't all follow the same pathway. There were actually 3 morbidity profiles that seemed very common. The first are their survivors. These are the people who had the onset of their first age-associated illness before the age of 80, and yet still managed to live another 20 years. And so it seems that they really have a great resilience to age-associated illnesses. The next group, comprising 45% of our centenarians, were the delayers. So the onset of their first age-associated illness happened at or beyond the age of 80. And to put this into perspective, that was well beyond their median life expectancy for their birth cohort. So most of their peers had actually already passed away, and they were just getting their first age-associated illness. And then perhaps even more exciting are this group of escapers who reach the age of 100 without the onset of one of these age-associated illnesses. And they do make up a much smaller proportion of our centenarians, only 13%. But taken together, the delayers and escapers really seem to have a resistance to age-associated illnesses that we want to learn about. So we're not trying to get everyone to be 100. We're trying to get everyone to be able to be resilient or resistant to these age-associated illnesses. And another interesting thing that I'll point out here is that 85% of our centenarians are female. So the men are far fewer, but they are twice as likely than the women to fall into that escaper category. So they do seem to be more robust, even though they are much lower in number. And the delayers and escapers that I just mentioned really fit the notion of compression of morbidity that was put forth by Jim Fries, who used to be at Stanford University. And he stated that if life expectancy is fixed and we can delay the onset of disease, then we'll see a shortened amount of time that people are living with disease or morbidity. And so if you take this example here of a lifespan of 79 years, and we have a disease onset, we'll say of 60 years, then the person is living 19 years with a disease. But if we're able to delay the onset of age-associated illnesses due to increased preventative medicine, say delaying it to the age of 70, Now you've gained 10 disease-free years and shortened the amount of time that you were living with a disease. And that is called compression of morbidity. So what do we see in our centenarians? They have a very long lifespan. So does this translate to compression of morbidity, or are they just adding more years of life with disease, which is not really what most people are hoping for? And you can see in these survival curves here, we had a control group, a younger control group shown in black, nonagenarians shown in red, centenarians in green, and then those older and older centenarians, the semi-supercentenarians and the supercentenarians in blue and teal. And you can see that the survival curves show that the oldest centenarians have the latest delay in the age of onset. So our controls on average had their first age-associated illness at 71, but our centenarians didn't have their first until age 95. And then those supercentenarians, their first age-associated illness was at 109. But does this translate to actually a shorter percentage of your life living with a disease? And here we see that the controls have an almost 18% of their life they're living with a disease. But centenarians, it's down to 9%. And again, those supercentenarians really display this compression of morbidity. They are only living 5% of their lives with an age-associated illness. But what about functional abilities? So we had seen in our early studies that 90% of our centenarians were living disability-free independently into their 90s. And when we looked at activities of daily living across time, we see that the oldest centenarians, those supercentenarians age 110 plus, really are delaying the onset of functional impairment or maintaining independence longer than even younger centenarians. And we had seen that 50% of our centenarians were cognitively healthy within 1 year of death. So using a cognitive screener, we looked at this over time. And again, the supercentenarians, the most extreme of our centenarians, are compressing the amount of time that they're living with cognitive impairment. So our supercentenarians in particular not only show compression of morbidity, but also show compression of disability. And so it really shows us that they are a powerful group in particular, although I would say the centenarians are also quite informative for learning about healthy aging. So instead of studying disease with our centenarians, we're actually studying the opposite. How do you avoid disease? And people always ask, so is the health of centenarians due to a genetic predisposition to healthy aging, or is it due to good lifestyle habits? And much of what we know about how much our lifestyle habits contribute to longevity comes from the Seventh-day Adventist Health Studies, where this is a group of people who live throughout the United States, but they share a lot of healthy habits that are part of their religious doctrine. So they're vegetarian and they eat in moderation. Their religious doctrine tells them to take care of their body, to get regular exercise every day, to not use tobacco. And they participate in religious and family activities with a lot of social connections. And on average, they live 10 years longer than the rest of the US population, just by having those healthy habits. And of course, we know there are other important determinants of survival. Years of education and socioeconomic status are associated with living longer. And we know that heritability of longevity is relatively low, about 20%. But those figures come from twin studies that were done in cohorts where the oldest subjects were in their 80s or 90s. So we're really talking about the heritability of average longevity. And in our studies, we're looking at heritability of exceptional longevity, which is a whole different ballgame. And we have a lot of evidence in our studies that there is a strong genetic basis of exceptional longevity, stronger than what we see for average longevity into your 80s or 90s. So there's definitely familiarity. In our studies, we have many examples of pedigrees that include many centenarians. We enroll a centenarian and they tell us how their mom lived to 100 as well. Or we see a centenarian who has multiple siblings living to age 100. And again, I point you to this picture of Sarah Knaus, who lived to 119, and her daughter lived to 101. We also know that siblings of centenarians have an 8 to 17 times greater chance of becoming a centenarian themselves than the general population. And we've seen that the offspring are following in their parents' footsteps. They also are healthy agers. They have markedly decreased risks for heart disease, stroke, diabetes, cancer. And another, or maybe the opposite side of the coin, some work by other centenarian studies have shown that it doesn't seem like lifestyle habits are playing as great a role in one's ability to reach age 100. So Nir Barzilai and the longevity studies at Albert Einstein have shown that centenarians didn't differ in their levels of physical activity or diet from the general population, again pointing to the greater role that genetics might be playing with our centenarians. And work led by Paola Sebastiani looked at the genetic contributions or genetic determinants of living to these extreme ages. And she found that actually a signature of 281 SNPs could distinguish between our centenarians and healthy controls. But the interesting thing here is that these 281 SNPs could correctly identify people who would or would not live to age 95, 60% of the time. But it was much better at differentiating people who would live to the most extreme ages, 106 plus. Now we had 85% sensitivity. So showing once again that the most extreme ages are the most informative. They have the biggest genetic component here. And what kind of genetic variants do we see? So we hypothesized that with extreme lifespans, we would see that they would have lower disease-associated variants because they were delaying or escaping age-associated illnesses, and that they would have an enrichment of protective variants. But our study and several others have seen that longevity in centenarians and also in familial longevity shows that actually they don't lack the disease variants. And this is perhaps even more exciting because it suggests that they must have protective variants, strong protective variants that are preventing them from getting diseases for which they have risk variants. And then I don't think it's all genetics. Even just meeting with lots of centenarians, it does seem like there are psychosocial factors that contribute to longevity. And a big one that I've seen is a positive outlook. They really are happy to have lived to age 100 and they are enjoying it. And you see here on the bottom, these are actually two sisters, age 100 and 103, from a great photo shoot that was done by Boston Magazine. And Agnes said, the only thing I can say is don't dwell on the bad things. Always think of the good things and the exciting things in your life. It's not going to do you any good to dwell on the bad things. Really pointing to perhaps stress mechanisms. They've lived through a lot of good times and bad times and been able to deal with them and move on and stay engaged in life. But there are limitations to what we can learn from centenarians. It is very difficult to find the appropriate referent cohort for a centenarian. You really can't compare a 100-year-old to someone who's currently in their 80s because of significant cohort effects. Our initial centenarians were from the 1900 birth cohort and lived through things like the Spanish flu and world wars. And so to compare them to someone born after that really would not be appropriate. And we have tried to look at longitudinal cohort studies, and some of them do have centenarians because they follow a whole cohort over time. Some of those people will become centenarians. but centenarians are so rare that the number of centenarians in these studies is usually too few for meaningful comparisons. Another thing about studying centenarians is that it can be difficult to tease out what protective factors got someone to age 100 versus what are changes that we are seeing that are more related to end of life or terminal decline. And then we also have a very limited ability to study earlier life factors that may have contributed to getting to age 100. And in my case specifically, I'm interested in which factors help them maintain good brain health. And so we follow centenarian offspring as well. And as I mentioned earlier, we see that they also are healthy agers. They're following in their parents' footsteps. They have lower mortality compared to their peers, reduced prevalence and incidence of age-associated illnesses. And then we also can follow them over time. So here we administered a cognitive screener every 2 years, and we could look at how long they remained cognitively healthy. And we saw that our centenarian offspring were 46% less likely to be impaired at baseline, and also 27% less likely to become impaired over 8 years, suggesting that they have a more optimal brain aging pattern. And studying centenarian offspring has also given us the opportunity to study midlife factors that are associated with longevity. And here I show you some examples of psychosocial factors that we see in the centenarian offspring. So they are low in neuroticism, and so they don't experience a lot of stress about things that have gone wrong in their lives. And that sounds very similar to what I already told you about the centenarians, not surprisingly. And then they rate high in extroversion. So they tend to be outgoing, positive, and active. And this relates to having more relationships, bigger social networks that allow you to increase cognitive interactions and have improved safety nets. And then we also looked at purpose in life in our offspring, because this is a facet of well-being that is related to having goals and a sense of direction and meaning in one's life. And it has been related to lower mortality and morbidity in studies of older adults. And there was even a study where higher purpose in life, people with higher purpose in life were 2 times more likely to avoid Alzheimer's disease. So I wanted to see if maybe this was something that was playing a role with our centenarian offspring. And indeed, they had almost a 2 times greater odds of high purpose than their own spouses, and an almost 3 times greater odds of having high purpose in life than demographically matched participants from the health and retirement study. So indeed, these may be playing a role in these healthy aging trajectories. And so over the years, we've really gathered a lot of evidence from our study and other centenarian studies from around the world that exceptional longevity is associated with healthy aging. These are strong models of healthy aging. And so we've moved our centenarian studies into a new phase of what we call deep phenotyping. And these projects are led by Dr. Tom Pearls and Dr. Paola Sebastiani, and it's through 2 projects funded by the NIA, the Longevity Consortium's Centenarian Project and the Integrated Longevity Omics Project. And between these 2 studies, we are enrolling 1,850 centenarians and their family members and some controls. And we're collecting lots and lots of data on them. So we administer a wide range of questionnaires that you can see here, ranging from medical histories to a food frequency questionnaire, questionnaires about health habits, about occupational and driving history, well-being, lots of items. And then we are collecting biospecimens, including fasting blood samples, as well as stool samples, which we will be looking at in conjunction with the food frequency questionnaire data. And I should point out that we actually have someone who you probably know well, Andersen, who is helping us look more in depth at this food frequency questionnaire and how healthy eating index patterns of food consumption might be playing a role in longevity. And then from the blood samples, we are collecting a wealth of omics data from whole genome and whole exome sequencing, gene expression, all kinds of omics, so that we can really look comprehensively at what is contributing to these healthy aging trajectories and how can we learn from them so that people living to any age can see this same compression of morbidity and disability. Now, beyond the questionnaires and biospecimen collection, we actually assess centenarians in a way that has never been done before. So for each participant, we conduct a hybridized Zoom-based assessment for health and cognitive function. We send each participant a box of equipment containing a touchscreen tablet that is both Wi-Fi and cellular capable. And this allows us to present our test stimuli in a very standard way. And since some of our participants have very limited experience with computers, we have actually pre-configured all of the tablet and Zoom settings so that aside from turning on the tablet, our staff can remotely control all functions of the Zoom assessment and tablet. Also included in the kit are a secondary webcam and wireless headphones, a digital thermometer, a blood pressure monitor, a dynamometer to measure grip strength. And then by video conference, we walk the participant through how to set up all of the equipment and perform the assessments. And we also collect a layer of high precision digital data. So on the bottom right of the screen, you'll see the accelerometer that we send to the participant, and they wear it for a week. And this captures more than 100 parameters of physical activity and sleep. And we also do a neuropsychological assessment, which includes digital capture of spoken and written responses with the digital pen and digital voice recorder that just popped up. And we switched to this Zoom-based assessment because of COVID. We had originally planned to go see everyone in their homes and then had to pivot. And actually, this has worked out really well for us because it allows us to assess people over multiple days, since among our centenarians, fatigue can be interfering with our assessments. And it also allows us to reach people in any part of the country, not just those who we could easily travel to by taking flights to major cities. We actually can send this box out anywhere that we need to. And just a little bit more detail on the neuropsychological assessment that we do. It's actually about 90 minutes long, and it goes well beyond the use of cognitive screeners, such as the mini mental state examination that you see at the top. We do administer that just as a crude global measure of cognitive status. And really, it's because that's the measure that has been the most used in centenarian studies. But cognitive screeners such as these were really only developed to screen for possible dementia. They really were never meant to be a measure of cognitive function per se. And we want a more comprehensive look at cognitive function in several different domains that I have listed here. One, so that we can better detect mild cognitive impairment, which screeners are not good at doing. And so that we can better understand which specific cognitive functions change over time, remain stable, or even get better in normal cognitive aging and at these extreme ages. And as I mentioned, all tests have this digital layer of data collection. So any written test, the participant uses a digital pen, which I'll tell you a little bit more about in a minute. And then any spoken response to a test is captured with a digital voice recorder so that we can look at very subtle differences in cognitive test performance well before someone starts scoring poorly on a cognitive test. And so what do we do with all of these neuropsychological data? Well, we actually can cluster participants to group them by those that have similar cognitive profiles. So we took all of the cognitive tests that we had in one study, and grouped participants whether they had similar high or low performance on each test. And so you can see the group on the far left performed poorly in all of the tests. The group on the far right performed very well or above average on all of the tests. And in between, there's a mix. So there's no one signature of cognitive function. Each individual has lifelong strengths and weaknesses in different domains of cognitive function. Moreover, different neurodegenerative diseases elicit different cognitive profiles. So with Alzheimer's disease, we more typically see declines in episodic memory, whereas a different neurodegenerative disease like primary progressive aphasia, we tend to see more prominent declines in language function. So it really is important to differentiate different cognitive functions rather than just looking at a global cognitive screener or even compiling composite test scores from performance across all these tests. You really lose the nuance that is so valuable from a neuropsychological assessment. And what we did in this study is we actually took those cognitive profiles and annotated them with different clinical variables, polygenic risk scores, and blood biomarkers to understand what might be underlying these different cognitive performance profiles. And we found that some clusters were associated with higher mortality, some with cardiovascular disease, and some with dementia, showing that we actually can capture intra-individual differences in cognitive performance that might be related to underlying health conditions. And now I'll circle back to this digital pen, which we use, as I said, to get a very precise measurement of cognitive behaviors. So these digital tools allow us to leverage the extreme precision that digital data can capture, that a human could not capture. And these may be more subtle signs of early cognitive impairment. So if we take the digital pen, it writes on a piece of paper and it captures time-stamped X and Y coordinates every 12 microseconds or milliseconds, something like that. And we can actually use those data to extrapolate how much time did the person spend writing. So the pen was on the paper and drawing. How much time were they holding the pen and thinking about their next move? And we call that thinking time or cognitive processing time. And then how does their performance change over the course of a test that has a continuous demand? And so if you take, for example, the clock drawing test, it's a widely used test of cognitive function. And you can look at these clocks. We tell the person to draw a clock, put in all the numbers, and set the hands to 10 after 11. And it's very obvious in this picture here that someone who is healthy has a different or a more intact clock than the person shown here with Alzheimer's or the one with Parkinson's. But now if you take two clocks that are correct, how can you differentiate which of these two people will go on to have cognitive impairment? And this is where we need to dive deeper into performance on these cognitive tests. And the clock drawing test is one of the most commonly used avenues right now for understanding how these digital metrics that we get from a digital pen can be informative, well beyond just whether or not the person gets the clock correct or not. So we can look at things like clock diameter, how many pen strokes they used, what type of planning did they use? Did they put in the 12, 3, 6, and 9 first to help them with the spatial arrangement? Latencies. So now we can actually look after they draw the outline and put in all the numbers, how long do they pause while they're trying to think about how to set the hands to 10 after 11? And that's actually one of the most meaningful characteristics of these digital data. These latencies have been associated with mild cognitive impairment, differences in brain volume on neuroimaging, and ApoE allele carrier status. So it's a healthy or correct clock, and yet there are underlying very subtle differences between people who are going to go on to have cognitive impairment and those who will not. And the earlier that we can detect who will become impaired, the earlier we can intervene. And actually, the clock drawing test has been adapted by a healthcare company, and it's currently being used in many clinical settings as a more sensitive cognitive screener for cognitive impairment above or in addition to things like the mini mental status examination or the MOCA. And then just another example of what we've done with digital pen data is that we looked at performance patterns on the digit symbol substitution test. So this is a coding test where we give participants a key where numbers are paired with symbols. And then we give them a grid with empty boxes with a number in the top of the box and a blank box on the bottom. And they have to put in the correct symbol that matches each number as quickly as they can for 90 seconds. So we use the digital pen data to separate out the total completion time into how much time they spent thinking or looking back up to that key to find the correct symbol. And we saw different patterns of change. So whether people got faster or slower over time or stayed consistent in the amount of time that they needed to look and find that the matching symbol for each item. And interestingly, the ones who got faster or slower over time, it was associated with poorer episodic memory. So this is not considered to be a test of memory, and yet we found strong associations with episodic memory, suggesting that the digital data are capturing something that we are not capturing in the overall test score. But then we also looked at writing time. So just looking at the amount of time that the pen was on the paper, drawing in each symbol. And participants who had a slower time throughout the 90 seconds, they got slower and slower. That was associated with poorer physical function, things like worse grip strength or slower gait speed. And so a really interesting thing here is we could use these digital data to say this person has a very slow score or a poor score on the digit symbol substitution test. Now we can tease out what are the cognitive components to test performance and what are the motor components to test performance. And they're very subtle differences, but they do seem to be meaningful. And so now I want to switch gears a little bit, and I'm a behavioral neuroscientist, so I'm particularly interested in our centenarians who are cognitively healthy as an extreme of cognitive aging. But there's another model for studying extreme cognitive aging, and these are the cognitive superagers. The most well-known study is the Northwestern Superager Study, which is now led by Emily Rogalski. And they saw that if you look at episodic memory performance by age, there's a general average decline with increasing age. Some people have episodic memory deficits due to underlying neurodegenerative disease, and those are more significant declines. But even cognitively healthy older adults do see some minor declines in episodic memory with increasing age. But there's a lot of heterogeneity. So you can see among the 80-year-olds in this graph that some of them, the majority of them are falling in this gray zone. Some are below it, indicating that they have a significant cognitive impairment in episodic memory. But there are these people at the very top who are performing just as well or even better than 50-year-olds. So their study focuses on 80-year-olds who are performing on episodic memory tests at the level of someone 3 decades younger, a 50-year-old. And the reason that we're so interested in cognitive superagers is because of all the evidence that the Northwestern Superaging Study has produced, showing that there is something different going on in the brains of these cognitive superagers. So in panel A on the upper right, we see that when you look at older adults, say 80-year-olds, average 80-year-olds, there's a lot of cortical thinning in comparison to average 50-year-olds. But when you look at panel B on the lower right, this is a comparison of the 80-year-old superagers to average 50-year-olds. And there are no thinner areas of the brain among cognitive superagers. And actually, they even have a thicker anterior cingulate than 50-year-olds, people 30 years younger. And in addition to these striking results with cortical thinning, not surprisingly, this cognitive superagers have thicker brain cortex than their same-aged peers with average memory. And then we also see differences in functional connectivity and slower rates of change and atrophy over time. So these cognitive superagers also seem to be on a different healthy cognitive aging trajectory. And this really struck me as something similar to what has been coming out from centenarian studies. So this is some information from the 100+ study in Netherlands, which is led by Henne Holstege, and they study only cognitively healthy centenarians. And they have a brain donation study where they have looked at 40 brains from cognitively healthy centenarians, and the vast majority of them had little to no atrophy, even though they've lived to age 100. And then a case series of centenarians from 3 different brain banks, interestingly found that even though advanced age is the #1 risk factor for Alzheimer's disease, 1/3 of centenarians are able to avoid it. So 21% had no amyloid plaques, and an additional 17% had low levels of amyloid plaques that would not qualify for pathological Alzheimer's disease. And so this led us to want to merge the extremes of exceptional longevity and cognitive superaging. And therefore, we started the Resilience and Resistance Against Alzheimer's Disease in Centenarians and Offspring, which we call RAGCO, where the overall objective is to find these centenarian and offspring cognitive superagers, gauge their cognitive resilience, and understand the underlying protective biology so that we can develop therapeutic targets that will help everyone maintain these optimal brain aging trajectories. And so coming back to that plot that I showed earlier, our offspring of centenarians, the ones that qualify as cognitive superagers, have that really exceptional episodic memory performance, well above what's average, not only for their age, but also at average or better to someone 30 years younger. And then we have this really exciting group of centenarian superagers, and you can see we're really jumping off the plot now because we're looking at 100-year-olds who have performance at the level of someone in their 70s and potentially even younger than that. And once we identify our cognitive superagers and we also study average performers, we want to understand what are the underlying differences in brain health that contribute to this youthful memory in older age. So we look at blood biomarkers to see if the superagers maybe have markers of lower inflammation. We look at neuroimaging to understand whether they have better structural integrity and functional connectivity than expected. And we do a neuropathological assessment after the participant dies to get a detailed look at their brain health. But this not only gives us the opportunity to identify factors that are related to cognitive superaging, Since we know that advanced age confers a higher risk of AD pathologic change, as shown in this graph here, and that AD pathologic change has a high association with cognitive impairment, our entire sample is cognitively healthy, and yet they have advanced age. So there's a disconnect somewhere here. And for those who have evidence of AD pathologic change from blood biomarkers, neuroimaging, or on neuropathological assessment, yet they don't have cognitive impairment, we say that they are resilient to Alzheimer's disease. They have the AD pathology, but they're coping better with it. They're maintaining good cognitive function, or potentially even exceptional cognitive function in the case of the superagers. Whereas for our centenarians, if they are not showing AD pathologic change in those above markers, then we term them resistant to AD pathology. So they have completely avoided Alzheimer's pathology. And it's important to distinguish these two, or to determine these endophenotypes, because they likely have very different underlying biological mechanisms, completely avoiding Alzheimer's pathology versus having mechanisms to deal better with it. And so just very briefly on the different types of markers that we're looking at to gauge resilience and resistance, we look at blood biomarkers that you see here. Our primary marker is p-tau217, which has a high correlation with both amyloid and tau PET imaging. So if the person has a high level of p-tau217, we call them resilient to Alzheimer's pathology because they have evidence of Alzheimer's pathology. And if they have low p-tau217, we call them resistant to Alzheimer's pathology. In terms of neuroimaging, we're looking at structural and connectivity data, and we're even doing very novel measures of blood-brain barrier integrity. We're, of course, looking at microbleeds and myelin integrity. So really trying to understand what's different about these brains of our cognitive superagers, but also how are they avoiding or coping better with Alzheimer's disease. And similarly with neuropathology, our centenarians have a high mortality rate, so often it can actually be easier to look at their brains through neuropathological assessment than through neuroimaging. The data are a little bit different, of course, but we look at a whole range of neuropathological diagnoses as well as quantitative outcomes that you see listed here, and even some more novel measures looking at synaptic density and blood-brain barrier integrity. And taking evidence from other centenarian studies, notably the 100+ study, they have found that 50% of their cognitively healthy centenarians are resilient to Alzheimer's disease. So they have higher levels of Alzheimer's disease pathology that should cause cognitive impairment, but it doesn't. So they're coping better with that pathology. And then 50% are resistant to Alzheimer's disease pathology. Again, despite having the #1 risk factor for Alzheimer's disease, they really have very low levels of AD pathology. And so far, we've had 2 brains come to autopsy from 2 of our centenarian cognitive superagers, and both of them had intermediate levels of AD pathology. And these are really 2 extreme cases of resilience to Alzheimer's disease that we believe that we can learn a lot from. And we're doing that through collaborations with several different groups of people. So we're looking at genetic pathways, underlying resilience and resistance, transcriptomic pathways by assessing RNA in peripheral blood and brain tissue. And then I, of course, am interested in the behavioral and environmental factors, which I didn't show here. And then in the translational arm of our study, we're also deriving induced pluripotent stem cells from these cognitive superagers so that we can grow out neuronal and support cells and perform functional analyses in a dish to see if they act differently in response to stressors like AD proteins. so that we can really understand the mechanisms that are protecting these superagers from Alzheimer's disease and other brain changes. And then we not only are testing the individual cell types, but also using 3D AD-in-a-dish models that combine multiple cell types at once to really allow us to look at these neural cells as a brain system. And just to sum up, there are many different studies I mentioned here, as well as some others that I didn't, and they all have a wealth of data. I hope I have impressed upon you the amount of data that we are collecting and the richness of these data. And we have expertise in our study, but everyone out there has additional complementary expertise. Our data and resources are shared at these locations listed here, and we are always welcoming collaborations as well. And then just a quick thank you to all of our collaborators who I actually couldn't even fit on this slide, our amazing participants, and our essential funding from the NIH and the McKnight Brain Research Foundation.
Good. Thank you very much, Stacy. That was a fantastic talk. And we do have questions in the queue. First question comes from Larissa Constantino, who thanks you very much for a very stimulating talk and says that she's curious whether centenarians speak several languages and whether they have lived in several countries.
So back when I first started with the study, we actually did have a lot of people who had immigrated from other countries among our centenarians, and several of them did not speak English, and so, or they spoke multiple languages. And I was very interested in the role of how bilingualism may have helped them maintain very good brain function to these very extreme ages. I didn't find any robust differences between the people who were bilingual or monolingual, but I think as our sample size grows even further, it's definitely an avenue we're looking into.
Well, I'm waiting for more questions in the queue. I was just curious regarding your studies looking at the resistant and resilient centenarians, and whether there's any plans at this point to actually look at diet and what role that might play.
Yes, so we do have people looking at dietary patterns among our centenarians, and we're also very interested in looking at the role of diet in conjunction with the data that we'll be getting from the stool samples. And there's a lot of evidence showing that the gut microbiome contributes to brain health and avoidance of Alzheimer's disease. And of course, things like the healthy diets, Mediterranean diets, are known to be related to keeping your brain healthy. So I would not be surprised if we find important correlates.
Well, thank you. Another question just came in and from Saidas, who says, kudos to you for putting together an excellent battery of remote assessments. This is a great model. In addition to the advantages, are there any limitations that you have observed?
There are some limitations. We weren't able to do all of the assessments that we would like to do remotely. So when we visit people in person in our other studies, we do things like gait speed and chair stands, and we really aren't able to do those remotely. We instead supplemented our assessment with the use of the accelerometer that we hope we can get some valuable data on gait speed and even things like stability in lieu of.
All right, I'm waiting to see if any more questions will pop into the queue in the next few seconds. And if they don't, I would again like to thank you for a really fascinating talk. And I really look forward to the work that's going to come out of this research. Thank you very much.